It's a privilege to hear those stories, and it's incredibly brave of both you and Pachyonychia to be so open and just to share that. It's really inspiring. I now invite Professor Edel O'Toole, Lecturer of Dermatology at QMUL, to talk about her involvement with the PC project.
Thank you. So thanks very much. So I've been involved with the PC project since about 2005, I think. And I've been part of their genetics team. We review cases monthly during that time. And these are my disclosures. I don't personally get any money. The university gets it all. So as Dave mentioned, the IPCC started in 2004. There were approximately 400 individuals worldwide who were involved, but I guess there was a small number that are very actively involved. As well as the steering committee and genetics committee, there's a research committee, which is led by Pierre Colombe, and they review requests for funding, define priorities. We set up a PC project grant protocol, and look for outside research funding both in and outside of the United States. And in the last year we published a research agenda in the Journal of Investigative Dermatology, so that was sort of a milestone in that it's there as an agenda for scientists who might be interested in PC, and also for policymakers, for grant-giving bodies, etc. There's a communications committee which is involved in social media communications. So I'm going to talk a little bit about the power of having a registry for PC patients, treatment ideas in progress, and a little bit of basic science research, not very much. So a really important paper was a paper by Liat Samuilov and Ellie Sprecher, where they looked at 815 patients from the registry, and they showed in that paper that 19% of cases used walking aids. So that's a very critical thing to be able to say to a research grant-giving body. This is a serious disorder that causes disability. Also, 26.2%, so around a quarter of patients, reported depression or mood change. Maura Pawlowski, who also works with Ellie, looked at PC and hidradenitis suppurativa, and a survey of the PC register revealed that about a quarter of patients with PC had hidradenitis-like clinical issues, and these were mainly patients with PC keratin 17. Zhang Tan, working with me, looked at neurovascular structures in PC. So we sent a questionnaire to the registry. We got 281 responses, and 62.3% reported neurovascular structures. And these were associated with significant pain in all PC genotypes. So in general, the worse the PC, the more of these vascular structures you have, which you can see here, I think. Some people actually mistake those for warts, believe it or not, dermatologists. Lloyd Steele, also working with me, looked at PC and itch. The pain is obviously the big thing, but patients are also itchy. We hypothesized this might be related to inflammation in the skin, and there are also known changes in the nerves in the skin. So 68% of patients reported itch, about half in the previous month. The sites affected were under the plantar calluses, not all that surprising, and around the calluses, and also around cysts for patients who have cysts. What precipitated the itch? Wearing shoes for a long time, walking, or heat. Heat is known to make PC pain worse. Patients complained of tickling, burning, prickling, tingling, so various sensations. I have observed that patients with PC possibly have something called secondary erythromelalgia. This is where you get redness of the feet with increased skin temperature and burning pain. It's aggravated by warming and relieved by cooling. There's a primary type, which is genetic, and there's a secondary type. So for example, secondary erythromelalgia sometimes occurs in diabetes. But in lots of other conditions, Olmsted syndrome is a good genetic example where you've got mutations in TRPV3. So I observed this because I had seen quite a lot of patients for trials, and I noticed that some people just had very red feet, whereas other patients didn't. So we devised a questionnaire amongst the genetics committee. We sent it to all the patients on the IPCC database, and we looked at the erythromelalgia diagnostic criteria, and we invited patients to send photographs of their feet for review when they're red. We basically found that over a fifth, almost a quarter of patients, self-reported symptoms, including all the diagnostic criteria for erythromelalgia, and these patients had significantly higher average pain score. So this is something like a vascular instability within the skin that contributes to the pain in PC. And there was no significant differences between the 5 PC keratin mutation groups. Repurposing treatments, I'll just talk about briefly. One of the first things that was tried was inhibiting mTOR signaling. Rapamycin, also known as sirolimus, is an mTOR inhibitor. It's likely to work in several ways. 1, by inhibiting mTOR, 2, by decreasing proliferation or cell division, 3, by decreasing angiogenesis, and the McLean lab in Dundee found that it directly targets keratin 6A mRNA. And when oral rapamycin, when cells were treated with oral rapamycin, there was a reduction in keratin 6A, and oral rapamycin was given to 3 patients who actually responded quite nicely in terms of being able to walk more, but they all got diarrhea. Subsequent to that, a company called Palvella Therapeutics developed a gel preparation called Qutoran rapamycin 3.9%. There was a phase 2 trial which was very promising, and then there was a phase 3 placebo-controlled trial which unfortunately did not succeed in its primary endpoint. So patients who received drug did no better than patients who received placebo. However, 30% of the patients who had placebo had relief in pain, so there was a big placebo effect, which is always a problem in pain trials. Inhibiting epidermal growth factor receptor signaling. So the epidermal growth factor receptor is an important receptor on keratinocytes, and it's important in keratinocyte proliferation and signaling. And Alan Hovnanian showed in Olmsted syndrome, and initially, where you get variants in TRPV3, that erlotinib can be used to treat this condition. So here you see patients at baseline and after 10 weeks of erlotinib. So the epidermal growth factor receptor and TRPV3 are closely linked in keratinocytes and skin. Erlotinib, for those of you who do not know, is a cancer treatment which is used in metastatic cancer, and it inhibits the epidermal growth factor receptor. It has quite a long side effect profile. This may be because it's being used in cancer patients who have metastatic disease, who are obviously very sick. But it does seem to help neuropathic pain. And 6 PC cases have been reported to date. Side effects include acneiform eruptions and other rashes, hair loss, and conjunctivitis. Alain and Justine Bassé in this very elegant paper showed that TRPV3 is overexpressed in PC callus, as are the EGF receptor ligands, epiregalin and TGF-alpha. Going forward, topical erlotinib preparations may be in clinical trials soon, so Palvella has a topical preparation which they're planning to use in EB simplex, but they may also use it in the future in PC. And Solgel also have a topical erlotinib preparation. There are topical TRPV3 inhibitors in a phase 1b trial. this is being sponsored by an Israeli company called Kamari Pharma, and they also have an oral TRPV3 inhibitor, which is a different compound which they hope to use in trials. There are also other EGFR inhibitors which are not yet off patent, and they may also be suitable. Thomas Magan has done some nice work in EB simplex, where he has used keratinocytes with mutations in keratin 14. And basically, when you've got a keratin mutation, you get these aggregates or clumps of keratin within the cell, and he has found some compounds that seem to reverse the aggregates in EB simplex cells. Nucleic acid therapy or RNA therapy. So there are various possibilities. This slide is taken from a neurology journal, actually, where neurology is probably the most advanced with regard to gene therapies. Antisense oligonucleotides are a possibility. siRNAs have been tried, but there have been problems getting them to penetrate into skin. And the other possibility is adeno-associated viral vectors. Gene editing is clearly the holy grail, and there are some efforts in this space. So a research grant was awarded to Marta Garcia Diaz from Madrid, who's working on gene editing for PCK6A. Basic science. David Kessel and Diana Blayton are doing some spatial transcriptomics, looking at genes that are expressed in specific areas of the skin in palmoplantar keratoderma and in normal palm. Pierre Coulomb has done a huge amount of work on keratin's basic cell biology. He's done some interesting work in the last couple of years on keratin 17, showing that it can go to the nucleus, and this may affect how cells divide and how they respond to DNA damage or stress. Sonia Lehman and Akin has shown that the mitochondria in PC skin cells do not turn over normally. We don't understand what this means, but the cells are tired, so there could be more work in this area. So in summary, there's lots of research going on. The registry gives huge amounts of valuable information, and there are better treatments coming. And from my perspective, it's been an absolute privilege to be involved with the PPC project and the ichthyosis support group. I see Mandy looking at me. So, I learn a huge amount from patients, at patient support meetings, at the genetics meetings. I've learned a lot from Ellie Sprecher and Alan Hovnanian. We all learn things from each other, I think. So it's been amazing. It's very powerful. I call on Dave now, who's going to close.
Thank you, Edel.
Just once again, this combination is powerful, and I know from the PC project, the work that the scientists and the doctors and the researchers do is just invaluable. We just completed our patient support meeting yesterday. It was a chance for the scientists and doctors to see many patients, and for the patients to see many doctors and scientists, and I think that was interesting. They learned from each other. In fact, you've heard of ground rounds. I called it the reverse ground rounds because they had 4 doctors going around talking to patients, and afterwards I said, how are the doctors? In the PC community, they carry each other. We carry each other. It's a very supportive, collaborative community, physically, emotionally, scientifically, in every way. We found that being together, it makes us stronger. We need each other. And once again, it was founded on love from a grandma for her grandchildren, but that's what continues to drive us. Vision is eliminating that pain and those who suffer from PC, the isolation and the embarrassment. It continues to be driven by love, but we're also guided by science. It's not all fluff. We have to have that science, that research, that technology to find the cure. And with that, we thank you very much for allowing us to be